So today we will be investigating osmosis process in both animal and plant cells, and we learn how to describe what will happen to the cell when the cell is immersed in a solution of different water potentials. So this is called describe. Okay, so we will learn how to describe what happens to the cell. So usually describe is what you can see, all right? And then also we will learn how to explain, that means why the cells behave like this when it's immersed in certain solution of certain water potential. So you need to explain and what are the points that you need to use to explain the process. Okay, so that's describe and explain for today's lesson. Okay, so here you can see that first we start off with the solution, right? And the solution has got a certain water potential. So this is an external solution and this is internal solution. So usually I'll prefer to, if you check your notes, I will have actually changed it, check and see. External environments, okay, external environment, okay, or extracellular fluid. I prefer extracellular fluid, okay, and this is intracellular fluid, okay, external and internal environments of the cell, okay, with respect to the cell, external and internal environment with respect to the cell, or intracellular or extracellular or intracellular fluid. Okay, first case is scenario is number 1. The water potential, okay, psi is the water potential of the extracellular fluid and intracellular fluid is the same. So both of them are the same. So let us pick a value to represent the water potential. So let's say, for example, external water potential is negative 1000 kilopascal, and internal fluid water potential is also negative 1000 kilopascal. Okay, so when the water potential on both sides of the membrane are the same, you can see that the rate of water molecules moving in will be equals the rate of water molecules going out, because water potential represents the ability of water molecules to move, which is also proportional to the number of water molecules, right? Number of water molecules proportional to, okay? So when I've got number of water molecules on the left side and on the right side are the same, okay? And they are inside the same temperature. That means the kinetic energy is the same, the number of water molecules are the same, so the probability of water molecule moving from left to right and right to left is the same. So therefore, they are moving from left to right and right to left, but overall, there is no net water movement. Okay, because number going into the cell and number going out of the cell is the same. So we say that there is no net water movement. Okay, so the red blood cell will look normal. So this is a description. It will look normal. Okay. Now, as for plant cell, it's also the same. Plant cell, there's also no net water movement. So the plant cell will look normal. So you need to draw the plant cell that looks normal and label all the structures. Okay. Nuclear, cell wall, cell surface membrane. Tonoplast, please remember, tonoplast is the membrane of the vacuole. The membrane that surrounds the vacuole is called tonoplast. You must remember this one, huh? Okay. Then the vacuole. And also remember, you have learned vacuole in chapter 1. You need to know what is the content inside the vacuole. Because I saw a past year question asking a student, 2 marks question. What is the content inside the vacuole? Right? Plant vacuole. So salts or mineral ions and sugar, also cell sap is acceptable. Cell sap. Okay. Right, so that is the first case when external and internal environment has got the same water potential. Now let's look at the second case, right? Case number 2. Case number 2, I have changed the water potential. So water potential in extracellular fluid is -500 kilopascal. Intracellular fluid is -1000 kilopascal. So you read these two values, -500 versus -1000, right? -500 versus -1000. Which one is higher? Shannon.
Is it lower? Teacher, can you repeat the question again?
So when you read the reading, right, the value -500 kilopascal versus -1000 kilopascal, which reading is higher?
The -500 kilopascal.
Are you sure?
Yeah.
Okay, don't only read from what I've written here. So you must know, right, based on the number, right? If this is if this scale here is 0, okay, so this is 0, okay. When you go forward is, let's say you go to the right side is positive, okay, positive. Then you go to the left side of 0 is negative, okay. So -500 will be located somewhere here. Sorry, where is my -500? Then my -1000 should be further down there, right? Okay, so -1000 is actually smaller than -500 because you know that if you are more familiar with positive, right? When a value increases, you know that it's bigger. So for negative value, this value -500 is actually bigger than -1000. So this is something that you need to pay attention to when we are talking about water potential values. -1000 is smaller than -500 according to the scale. You know, when you plot the graph and so on. Okay, that one is a common mistake among students, so you have to pay attention. So now the external fluid has got, okay, thank you, Shannon. External fluid has got higher water potential. Okay, so you've got higher water potential than internal fluid. So in which direction will the water molecules be moving, Joyce?
Into a cell.
Into the cell. So there is a net water movement into the cell, right?
Yeah.
Will there be water moving from the cell out of the cell? Shumin?
Can you repeat the question again?
Will there be water moving out of the cell?
No.
No. Okay. Now you got water molecules on the left and right side, right? Right. You got more water molecules on the left and the right side. Hang on, I draw again. I use blue color. Okay. So both sides also got water molecules, right? Which side got more water molecules? 30. Which side got more water molecules?
The left side.
Left side got more water molecule. Why? Why left side got more?
Because the pressure is higher.
Yes, because the water potential is higher. Okay, so left side got more water molecules. So all of them will have kinetic energy and they'll be moving, correct? And the kinetic energy actually depends on the temperature in the surrounding. So this cell is immersed in a certain solution. So the the temperature inside the cell and outside the cell will be the same. Okay, so therefore all the water molecules will have the kinetic energy, all of them. So there'll be water molecules that will be moving into the cell. Okay, and there'll be water molecules that will be moving out of the cell, because all of them got the same kinetic energy. Almost the same, cannot exactly same. Okay, so at any one time, there'll be molecules moving into the cell and molecules moving out the cell. So in both directions. Okay, but because left side has got more water molecules from the right side. So if you talk about probability, because you got more water molecules on the left side moving into the cell, you got less water molecules in the cell moving out. So therefore, overall, we say that to represent the movement, okay, this is the movement. There are water moving in. There are also water molecules moving out. But there are more, okay, I draw wrongly, hang on. Okay, there are also water molecules moving out. But there are more water molecules moving in than going out. So therefore, we say overall, based on this kind of movement here, when you consider all the water molecules moving in both directions, based on that water potential, therefore, I can conclude that there's a net water movement into the cell. Okay, but water molecules can move in as well as can move out. The only thing is that there is a net water movement. Net is a keyword. Net movement is into the cell, but movement is into and out of the cell. Understand? So can you see the difference between when you use the word net and when you don't use the word net is different? Okay, use the word net means overall, from left to right and right to left. Okay, so therefore, when a question asks you, when you say, oh, there's water molecules moving into the cell, you are not saying net, you're only saying this direction. Okay, and there's water molecules moving out, you're saying this direction only. But net means both directions being considered. Overall, there's more moving in than out. So using the word net in your description is very important. Without net, it's a different meaning already. Okay, so overall, there's a net water movement into the cell. Okay, so this is a first marking point. So this is a description. Remember, you need to describe, right? What do you see? Describe. So describe, what do you see? There's a net water movement into the cell, 1 mark. By osmosis, 1 mark. Why? So now you're explaining because, okay, there's one more point here. Just now it's described. So because down water potential gradient. Down water potential gradient. So I'm going to use psi to represent water potential. Okay, down water potential gradient. So this is another mark. So describe and explain. Okay, down water potential gradient. So describe again. So what do you see in the cell? The cell will swell and then eventually it will burst. So another word for bursting of the cell is called lysis. Cis is a process. Any word end with cis is a process. Lysis, lys means to break. So the cell will burst, okay, dispersing the cell contents. Right, so this is describe and explain. So this is for animal cell like red blood cell. What about plant cell? How to explain? So plant cell, how to describe and explain? There's net water movement into the cell. So you're talking about water movement, right? In plant cell, the water will move into the cell cytoplasm, and then it will also move into the vacuole by osmosis. Okay, eventually you'll move into the vacuole because vacuole has got mineral ions and glucose, sugar. So it will actually be a lower water potential inside there. So water will tend to go into the vacuole, all right? So the cell membrane and the protoplast swells outward. Now remember, protoplast is the living part of the cell, which includes cytoplasm and the membrane. Okay. So it will swell outwards and start to press against the rigid wall. So you imagine the cell, which is surrounded by the cell surface membrane. Okay, so originally the membrane is just touching the wall, it's not pushing against the wall, right? It's like you're standing near the wall, but you're not pushing the wall, but you're touching it. Okay, so now as the water enters the cell, right, the cytoplasm will start to swell outwards. So therefore, the membrane that's initially touching the wall will start to be pushing the wall already. So let's say, for example, I use green color to represent the cell wall. Okay, so this is the cell wall. So only one side of the cell to illustrate. And the red color line is to illustrate the cell surface membrane. Okay, so this is a cell surface membrane, originally touching the wall. So it's not pushing the wall, it's just touching the wall. So therefore, the psi P, okay. Now, because it's not pushing, it's not pulling the wall, so psi P is equals to how much? Generally,
it will be lower.
so if you are not pushing is positive, right?
Yes.
And if you are not pulling the wall, pulling is negative force, isn't it?
Yes.
So if you're not pushing and you're not pulling, you're only touching the wall, you're not pushing the wall. So what is the psi P value of the membrane onto the wall, the cell wall?
Is it 0?
Yes, it's 0. And what is the unit? 0 what?
Kilopascal.
Yes, very good. Kilopascal. Kilopascal. Okay, so 0 kilopascal. Okay, I have to write here, got no space. 0 kilopascal. Okay, so you can see that originally when the water enters the cell by osmosis in plant cell, right? So the cell starts to swell, okay? And then the membrane, originally, which is touching the cell wall, psi P is equals to 0 at that time. But as the cell continues to swell, because the water enters the cell, right? So when the water enters the cell, so let's say water enters the cell. So therefore, I don't use red lah. Let's use blue color better. Okay, use blue color. So as the water enters the cell, right? So the cell will start to be filled up with water, and the water will then press against the membrane already. So the cell will swell outwards. So when the water press against the membrane will press against the cell wall, isn't it? Correct or not? It's like you are pushing, let's say you got a balloon, okay? You blow up a balloon in a small box. So as you continue to blow the balloon, originally balloon is not touching the wall. So as you blow, the balloon becomes bigger and bigger, it starts to touch the wall, okay? It starts to touch the wall. Then you continue to blow. Then it will start to press against the wall already, right? Okay, just like the cell, water continue to enter. Okay, the cell will continue to become bigger and bigger, bigger. So initially it will touch the wall, after that it will press against the wall. So when the membrane presses against the wall, okay, what will be the psi P? What will be the psi P inside the cell? When you press the wall, you yourself, because we're talking about the cell, when you press against the wall, what will you experience? Tan Hui Min, now you can go and try and press the wall and feel for yourself. What do you experience? Do you experience a pushing force, a pulling force, or no force towards yourself? Tan Hui Min.
Pushing force.
Yes, pushing force, right? If you push the wall, how much the energy you push the wall is how much the energy you will experience back from the wall, right? Correct? So this is physics you learn with what? The third law of Newton, is it? Third law of Newton, right? Every action, there's a reaction with the same magnitude of opposite direction, right? Okay, Newton's third law, okay? So same thing in the cell, it will experience this Newton's third law. As the cell swells and the membrane touches the wall, and then the cell swells some more because water enters some more, and the membrane will push against the cell wall. The cell wall will push back the membrane in the opposite direction with the same magnitude. So you will experience the cell wall will push against the membrane. So inside the cell will be experiencing this pushing force. So when you experience a pushing force inside the cell, what will be the value of psi p for Shini?
More than 0 kilopascal.
Yes, very good. So it's actually more than 0 kilopascal because it's a pushing force, right? So therefore, we will say that the psi p experience inside the cell is more than 0 kilopascal, which means to say it's positive, right? It's more than 0 pascal. So will the water stops entering the cell? Megan?
No.
You mean the water will continue to enter the cell and the cell will continue to swell forever? Can the plant cell swell forever?
Yes.
Yes. Okay, you see, if you have got a box, right, and you put a balloon, let me see, you put a balloon inside the box and you try to blow the balloon. Okay, you try to blow the balloon. Now, when the balloon is that big, you continue to blow the balloon. Can it continue to expand? Let's say all four sides are already restricted by the wall of the box. Will it be able to continue to expand?
No.
No, right? If there's no box there, it will continue to expand like our red blood cell and eventually it will burst. But now its expansion is restricted by the wall that's surrounding it. So after you blow it until a certain extent, it can no longer expand already, right? It will stop there. So we want to know that when will the plant cell stop expanding, that the water can no longer enter the cell is when it will stop expanding, right? Right? The plant cell will stop expanding when the water can no longer enter. Now, when the water can no longer enter the cell anymore, there's no net water movement in the cell. So that means at that time, equilibrium is reached, right? So no net water movement anymore into the cell. So we say that equilibrium is reached, okay? So when you say equilibrium is reached, in terms of water potential, can you explain what you mean by equilibrium is reached? Samir?
Both water potential is equal.
Yes. Now we say that now the cell can no longer expand, right? The cell can no longer expand because there's no more net water movement into the cell. So when there's no net water movement into the cell cannot expand. So that means equilibrium is reached. That means at that time, we expect that psi, so the external water potential of the external solution, okay, the psi, okay, of the external environment. So I will say psi of the external environment, okay, external environment will be equals to the psi of the cytoplasm. okay, or internal environment. Okay, so when the cell stops swelling, we can say that psi of external environment is equals to psi of internal environment. Okay, so that's when the cell will stop swelling. So because the water potential is the same, so there's no more net water movement, so the cell no longer swells. So can everyone follow up to here? Okay, so let's read the description. So there's net water movement into the cell, to the cytoplasm, and then to the vacuole by the process called osmosis. So add one more point into your notes, because this is a very important point. It's 1 mark in exam. So add one more point down, water potential gradient. So this 1 mark exam, okay? So the cell and the protoplast swells outward and start to press against the rigid cell wall. So when it starts to press against the wall will push it back. So the psi p inside the cell will increase because the cell starts to experience the pushing force from the wall. So the psi p will start to increase. So when the outward psi balances with the inward psi, okay, when the outward psi balances with the inward psi, that means the water potential inside the cell and outside the cell becomes the same. Then we will say that there's no net water movement into the cell anymore, because water movement is governed by water potential gradient. Now, psi outside the cell and psi inside the cell is the same. So there's no more water potential gradient. Therefore, there's no more net water movement. Okay. So this one, we'll look at it later on in the next slide. I'll explain this one. Okay. So therefore, at that point, we say that the cell becomes turgid. That means turgid means what? It is inflated with water. It's just like the balloon inflated with air. Now the cell is inflated with water pressing against the cell wall. So this is called the cell is turgid. Okay. So you need to draw this where the cell is turgid. Water has entered into the cytoplasm and the vacuole of the cell. Okay, so this is to examine the water potential for 2 different cells. Okay, when the cells immersed in a solution with higher water potential. Okay, so now we look at psi of the environment. Okay, initially psi of the environment, external environment, is negative 500 kilopascal, and psi of the cell is negative 1000. So we already say that negative 1000 is lower than negative 100, and because of that, there's a net water movement into the cell. And the psi S of the cell is also negative 1000, because at that time, psi p is 0 initially. Because initially, the membrane of the cell is not pushing against the wall. It may be touching the wall, but it's not pushing against the wall. So there's no pushing force, there's no pulling force. So psi p is equal to 0. So because psi p is equal to 0, based on the equation of psi, okay, is equals to psi s plus psi p So at that time, because psi p is equals to 0, therefore psi is equals to psi s. So both these 2 values is the same. Okay, so that's why psi s inside the cell is equals to psi of the cell. It's negative 1000 kilopascal. Okay, so then we allow the water to enter the cell for some time. So eventually the cell becomes turgid because water, there's net movement of water into the cell all the way to cytoplasm and then to the vacuole. All right. So after 10 minutes, say for example, we allow the diffusion to happen for 10 minutes. Let us examine the water potential. All right. At that time, at 10 minutes. Water potential of the environment will still be the same, is negative 100 kilopascal. Okay, what about water potential of the cell? Question mark. Okay. What about solute potential of the cell? Question mark. What about pressure potential of the cell? Question mark. Okay. Now, water potential of the cell is calculated based on this equation. Psi of the cell is equal to psi p and psi s. So if you want to know the value of psi of the cell, you need to find out first what is psi s and what is psi p, and you calculate, you add these two values up, it will give you your value of psi of the cell. So therefore, we have to calculate, or we have to find out what is the value of psi p. Now, psi s is given here as -999. The reason here is because, why is it -999? Can anyone tell me? Filbert? From 1-1000, it has now becomes -999. Increase or decrease already?
Increase already.
Okay, why is it increased?
Is it because the water enter the cell, so the water potential increase, and then
Yes.
so the solute, what is it, potential increase as well?
Yes, very good. Very, very good. We already said that there's a net water movement into the cell, right? So when water enters the cell will have got more free water molecules, but the cell is so tiny, isn't it? You can't even see one cell with your eyes. You have to use microscope to see. It's actually very, very tiny. So a little bit of water enters. So it will change the water potential of a cell slightly only. So because a little bit of water enters the cell, all right? So therefore, the size of the cell definitely will increase slightly. Why slightly? Because it's very small, right? Okay, very small volume only, that's why. So therefore, because psi s is proportional to psi, when psi increases, psi s will also increase. So therefore, can you see the psi s here will increase, but very little. From -1000, it increased to -999, all right? So therefore, this is, I want to erase, hang on. Okay, so can you see that this value, -999, is actually bigger than -1000 because there's more water molecules inside. So psi s is higher, psi is higher. Now, as the cells, thank you, Filbert. Thank you, Filbert. So now let's move on. As the cells swell, it presses against the wall and the wall pushes it back. So it will experience a pushing force in the cell. Something is pushing the cell. The wall is pushing the cell. So psi p will be a positive value, right? Okay. Okay, so what will be the value of psi p? Now here, say for example, if the size of cell now is -500. Okay, why? Why is it -500? Who is it I've not called? Curti. Curti, why is the size of cell is given there as -500 now?
Because the size of the cell is now decreasing.
The size of the cell is not decreasing.
It's decreasing.
The size of the cell is decreasing.
The vacuole is decreasing.
The size of the vacuole, how can the vacuole decrease when the water enters? It must be swelling, 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 and eventually stop swelling, right? When the water can no longer enter the cell, it will stop swelling, right? The size will not decrease, okay? Can you see these two values are the same? Why is it these two values are the same?
Because the net flow of water movement from the left to the right and right to the left is now the same.
Yes, yes. So that's mean no net water movement when the cell is fully turgid, right? Fully turgid means water can no longer enter. It is maximum already, fully turgid. So when the cell, so the water continue to enter until the cell becomes fully turgid. So the moment when the cell becomes fully turgid, we know that, okay, now no more water can enter the cell. So there's no net water movement. So the moment the water cannot move, we can say that equilibrium is reached, isn't it? Okay, equilibrium. Equilibrium is reached, okay, between the external and internal fluid of the cell, right? Equilibrium is reached. So when equilibrium is reached, it means to say that the water potential in the external fluid is the same as water potential in the internal fluid, right? That's why equilibrium is reached. Our water cannot move anymore, right? There's no more net water movement, means equilibrium is reached, meaning the water potential outside and inside the cell is equal. That's why there's no net water movement. Can you see that? Okay, so now when you put negative 500 here, can you calculate psi p or not? Can, isn't it? So can you all calculate what is the value of psi p now? Who I have not called yet? I've called everybody, is it? Ong Shu Min. Ong Shu Min. What is the value of my psi p?
Negative 499.
Negative 499. Very good. Huh?
Don't have negative.
Don't have negative. Yes, cannot have negative. Cannot have negative, then what must you write?
499.
499. Hang on. You need to write positive 499 because when you write a negative value, you also need to write a positive for the value is positive. In this case here, okay? So it's positive 499 kilopascal when the cell is fully turgid. Okay, fully turgid. It means water cannot enter anymore. Okay, so this is the explanation to work out when the cell is fully turgid, what will be the psi of the environment, what will be the psi of the cell, what will be the psi s, and what will be the psi p. Okay, can you understand? So when the cell is turgid, psi of the cell will be equals to psi of the environment. But psi s of the cell, after we have calculated, is actually less than psi of environment, right? Psi s of the cell we have calculated is negative 999 kilopascal. Psi of environment is negative 500. So psi of s of the cell is less than psi of environment. Just read from this table and give the answer. Okay, it's very easy. Okay, so now this is the next example, which is the last example. So the last example is we immerse the cell into a solution which has got a lower water potential than the cell. All right, so negative 2000 is actually less than negative 1000 kilopascal. Correct? Correct or not? Correct? Okay. Negative 2000 is less than negative 1000 kilopascal. So because of that, there'll be a net water movement out of the cell. All right, so put an arrow to show there's a net water movement out of the cell. So let's describe. So there's net water movement out of the cell. So after that, you must, when you say movement of something in biology, right? First, you tell me the direction of movement, and then you need to describe it moves from where to where to where to where. So this is something that you need to say. Move from where to where through where. Okay, and then by what process? Okay, because of what? Okay, move from where to where is 1 mark. To where is 1 mark. By what process is 1 mark. Because of what is 1 mark. Okay, 4 marks when you're asked to explain and describe and explain. Okay, so first there's net water movement of the cell. Then from where to where to where to where? From vacuole through the tonoplast, through means across, okay? And then to the cytoplasm and through the cell surface membrane, across cell surface membrane, and then to the external environment, right? From where to where? Through this membrane, and then to cytoplasm, and through this membrane, and then to external environment, okay? So by what process? Shannon? Cepat sikit. Through what process? By what process? My Shannon is not responding. Okay, let's not waste time. Joyce. Yes, by osmosis, 1 mark, okay? 1 mark by osmosis. So you have to write there. So by osmosis is 1 mark. Can I say by diffusion? I don't like osmosis. I want to put diffusion, can or not? Charmaine? Can I say by diffusion? I don't like to put osmosis. After all, water moves by diffusion, right? Can I?
I don't think is that appropriate
Why?
because osmosis is only for water.
Okay, but my water in the beaker is moving by diffusion.
Yeah.
So when is it I use diffusion and when is it I use osmosis for water? Can water diffuse or not?
Yeah, I can.
Can, right? Water can diffuse on region higher to lower water potential, can, isn't it? Okay. So when is it I use diffusion? When is it I use osmosis? Why is it I use osmosis here? Why is it cannot diffusion?
I think diffusion is like for general because other substances can also move through.
Okay. When you use osmosis?
When it involves water.
When it what?
It involves water.
Involves water, yes. When it involves water, we can use osmosis, but water can also diffuse one. So in which condition that we use osmosis for water? Lipei?
When water is moving through the membrane of the cell.
Yes, when water when water diffuse across a partially permeable membrane, ppm. Okay, we say that water move across the membrane by osmosis. Okay, but however, when the water move up the xylem vessel from the root to the stem to the leaf, there is no membrane that will block it from moving because the xylem vessel cells are all dead cells. There are no membrane there. So why is it moving from the root to the stem to the leaf? Why is it water not moving from the leaf to the stem to the root and then to the soil? There must be a water potential gradient for it to move, right? So in that case, we cannot say the water move up the xylem vessel by osmosis because there's no membrane for it to move across. So in that case, we say that the water move up the xylem vessel by diffusion down water potential gradient. It is still down water potential gradient because we always say that when you want to say water movement, we can no longer use concentration to describe. We have to use water potential. If the water can move in one direction, definitely there's a water potential gradient. But because there's no membrane, then we say the water diffuse from a region of higher water potential in the soil to a region of lower water potential in the leaf. Okay, without crossing through membrane. But the moment in the leaf, when it enters the mesophyll cell, the water has to pass through the membrane. That process is called osmosis. Okay, so water can diffuse Water can also move by osmosis, diffuse when there's no membrane, osmosis when there's membrane. In both conditions, it must be going down water potential gradient one. Okay? So therefore, you need to know when you use osmosis, when do you use diffusion. Okay. Right, let's continue. So by osmosis, we say that in these conditions, by osmosis, because it involves the membrane. Okay, membrane of the tonoplast and the membrane of the cell surface membrane. Okay. So therefore, in plant cell, what happened? You imagine a cell that's turgid. Okay, let me draw again. Just now I used a box to represent. So let's use the box to represent. Where's my box? This box to represent the plant cell wall. Okay. And then the red color to represent the cell surface membrane of the cell. So this is my cell. It swells and presses against the wall, and the wall presses against it. It's fully turgid, okay? And then the water starts to move out of the cell. Okay, why? Because there's a water potential gradient, right? Outside, the external fluid has got a lower water potential than the side of the cell, right? Okay, side of the cell. I teach very slow. Never teach so slow before. Are you okay with my speed of teaching? Can give me feedback now, please. Is it okay, or is it too slow, or is it too fast? I don't think it's too fast. Is it okay, or is it too slow? Please give me the feedback so at least I know. Hey, can you please type there first so I know you're okay with the speed or not? Is it too fast, too slow, or just nice? Okay, comfortable, Very comfortable because you can understand as I move along, right? Okay, good. Okay, good. Januli? I didn't see you type God. It's okay. Very good. Shumin, yes, it's okay. So it looks like this speed is okay for everybody. Anyone not okay, please let me know. Okay, good. So you can understand it's good. Okay. Once you understand, go back and read again, then it's crystal clear already understanding. So now you see the cell that is turgid, you place the cell after it's turgid, you place it into a solution where the psi in the environment is lower than that of the cell. So because of that, there's a water potential gradient, right? So the water will move out of the cell now. So there's a net water movement out of the cell. So imagine the cell initially is like pressing against the wall. So as the water starts to move out of the cell, the pushing of the cell against the wall will start to drop. That means the psi p will start to drop as more and more water molecules move out of the cell, right? Because the balloon will start to shrink already, right? So therefore, the psi p will become less and less and less and less until one point where it's no longer pressing against the wall. So at that point where the membrane of the cell is no longer pressing the wall, it's only touching the wall. So it's not pushing, it's not pulling. So that time, the psi p is considered as 0 kilopascal. Okay? So the moment when the cell surface membrane stop pushing against the cell wall, then at that point, psi p is considered as 0. So this point, we've given a special name to the cell. The cell is experiencing incipient plasmolysis. Okay? The cell is experiencing incipient plasmolysis. So what do you mean by incipient plasmolysis? This is the starting point of plasmolysis. So initially the cell is turgid, and then now the water goes out of the cell, and then until the cell is no longer pressing against the cell wall. So psi p is exactly equals to 0. This is incipient plasmolysis, the starting point of plasmolysis. Because after this point, if the water continues to go out the cell will shrink, okay? The cell surface membrane, the protoplast will shrink away from the cell wall, okay? It will shrink away from the cell wall. So when the cells start to shrink away from the cell wall, then this we consider that the cell is undergoing plasmolysis already. Okay, so during incipient plasmolysis is considered as a starting point of plasmolysis, where psi p is equal to 0. As the water continues to move out from this point onwards, cell surface membrane will start to withdraw away or shrink away from the cell wall. Okay, so cell will then experience plasmolysis. So how does plasmolysis looks like? In the microscope, it looks like this. You can see that the membrane already shrink away from the cell wall. The cell surface membrane already shrink away from the cell wall. See not? So this is considered as plasmolysis. Okay. Can I ask you one question? Why is it that not every part of the cell wall shrink away? Sorry. Why is it not every part of the membrane shrink away from cell? See, this membrane is still touching the wall. How can it be this membrane touching the wall when other parts of membrane already shrink? Can you tell me why this membrane is still touching the wall? Megan? What pulling the membrane at this point so that it will not withdraw away from the membrane? Something is pulling it. What is that?
R interactions.
What interaction?
R group.
R group? No. I never teach that it's an R group. It's not protein anyway. Okay. Is there any part, the structure that we've learned in chapter 1, where the membrane of one plant cell is directly connected to the cell surface membrane of another cell? Charmaine?
I don't think I remember.
Okay. Who can remember? Please type your answer. Type your answer. Hello? Hello? I want to cry. This structure you learned in chapter 1. Hello? Something that's cytoplasmic stream across the cell wall, or cytoplasmic strand across the cell wall. What is it called? Cepat. My time is up already. Gilbert? Start with a P. P. Yes, someone type answer already. Yes. Mida Pamela. Huyen. Pamela is the name of a person, Pamela. Mida Lamela. Lamela is a Lamela. Lamela versus Pamela. Pamela. Okay. Lamella is a middle plate. It's a plate separating two cell walls. So it's not middle lamella. It's plasmodesmata, Joyce type plasmodesmata, Lao Huyen type plasmodesma. Did Lao Huyen type that? Okay. Middle Pamela. Okay, yes, it's actually plasmodesmata. Okay. Plasmodesmata. Right, remember? Across the cell wall of neighboring cell, okay, there's this cytoplasmic strand. How do I draw it? So this is a cell wall, right? Okay, this is a cell wall of neighboring cell. Okay, your middle lamella is actually right in the center of the cell wall one, separating the cell wall of two neighboring cells. And then your cell surface membrane, let's say I put it as a blue color now. So cell surface membrane here, right? You see not? Is connected to the cell surface membrane of neighboring cell. So it's being pulled by the cell surface membrane of the neighboring cell. So because of that, it may not shrink out yet to the center. So this membrane already shrink away, but this membrane may not shrink away because it's still connected and being pulled by the membrane of the neighboring cell. So that's why you can see certain point here, how come the membrane did not withdraw away from the wall? So did you ever ask yourself or not when you look at diagram, hey, how come this side I did not pull away from the wall one? How come the rest of the sides pull away already? Okay, so in this condition, we describe animal cell will experience crenation. That means the membrane will shrink and the cell becomes crenated, like folded like that. Okay, and the plant cell experience plasmolysis, and it will be flaccid. Okay, lack of water, flaccid. So here, let us look at the diagram to illustrate the normal red blood cell. Red blood cell that has expanded. Okay, eventually it will burst. No. This is a red blood cell that's crenated and red blood cell that's expanded. Okay, so next we're going to look at plant cell. So plant cell that is flaccid, normal, and turgid. Okay. Okay, learn how to describe the cell. So now we look at some example of a plant cell that is turgid. So if the cytoplasm contain pigments like onion, you know, onion have got a pinkish purplish color or magenta color. So it's because the cytoplasm got pigment. So if plasmolysis occur, you can actually see the pink color shrink away from the cell wall. So it's very easy to see. Okay. Now this one is turgid cell on the left and plasmolyzed cell on the right. Highly plasmolyzed cells on the right. Very easy to see as long as there's pigment inside the cell. Okay. Now this is a Elodia leaf cell. The leaf cell will contain a chloroplast, and chloroplast is green in color. So you can easily see the region is green in color. You can also see the cell surface membrane, which is this line here. Right? This line here are all the cell surface membrane. See not? Okay, these are the cell surface membrane. Right? Cell surface membrane. So you can see the cell surface membrane. Here is also highly plasmolyzed. And you can even see the nucleus clearly. This is the nucleus. How do you know this nucleus? You look at another cell also, same size, same shape. Okay, then it is the nucleus, and only one in every cell. So that's how you try to verify whether this is a nucleus or not. Okay, so that's all for today. We have covered looking at osmosis in plant cell and animal cell, learning how to describe and explain the process. Okay, so tomorrow will be an exciting day because we will be exploring past year questions for this chapter. So it'll be very, very interesting. I will give you the question today first, okay? And then we will discuss the question tomorrow. If you want to go through the question today, you can, okay? So I'll see you tomorrow and have a nice day and please stay safe. Okay, bye everyone.